Continuing now with mitosis and the cell cycle, we're picking up with slide 23. Looking back at interphase, which is happening before mitosis, remember, the cell's just carrying out regular activities, and you will see the appearance of the two centrioles pop on, indicating that the cell's getting ready to grow and divide, and the nuclear membrane will begin to what we call disintegrate. So here are two cells. You can see here on this one, C stands for centrioles. They're popping on close to the nucleus here. And the nucleoli, which are the dark spherical structures inside the nucleus, also begin to disintegrate just a little. As we progress with the cell cycle, in slide 24, we enter prophase. And one of the first things that's characteristic is that you'll see an aster, which is a type of cytoskeleton filament, appear. And this one's stained beautifully with fluorescent green. But the aster will appear. This is like a feather in a hat, I guess is a good way to think about it. And when you look at the actual cell and what's going on at the beginning of prophase, here are the centrioles that have popped on. The aster, which is the cytoskeleton, has also begun to appear. And this will turn into something called a spindle here in just a minute. Here's a nucleus, and you can see that chromosomes are now condensing out. So all this chromatin and DNA is uncoiling so that you can begin to actually see our chromosomes in the nucleus of the cell. In late prophase, you will see that the chromosomes have condensed out enough to see them. They look like an X structure, and there's like a midpoint, a center dot, if you will. And as this spindle comes down, and you'll see it coming down right here. Here's the centriole, and look real close. Spindle. This is the spindle. They're fibers. They will attach themselves to the middle of each one of these X-like chromosome structures. And where they do, that midpoint is called a centromere. You can also see it in some books as a kinetochore. They'll say it either way. So the spindle attaches to all the different chromosomes. It's going to act like guide wires, is a good way to think of it. In slide 23, we progress to the next phase of mitosis called metaphase. Meta means middle, and the chromosomes shown here in red are clustering straight through the middle of the cell, and that's how you can remember meta. For example, what do you have on your meta right now? Are you wearing a belt by chance? There is an enzyme called separase that triggers the separation of these chromosomes in the next phase. What does that word separase sound like? Sounds like what? Separate, doesn't it? Which is exactly what it does. Note, just check this out real quick. If you don't know what a centromere is from the previous slide, take a moment now to look it up in your glossary or to Google it.